The microscopes are of various types, and most popularly known microscopes, that is the simple microscope and compound microscope, what we are using in the laboratory. Apart from that, very popularly known as the electron microscope. And whatever the diagrams and other things regarding cell and other, what we are studying, it is electronic microscope images what we are studying here. Now, first of all, what is simple microscope? Simple microscope by which the materials what we are observing on the stage is little bit bigger and thicker, which are not clearly seen by the naked eyes. Those things we are observing under the simple microscope. And the things which are not clearly seen by the naked eyes, those materials we are observing under the compound microscope. And in laboratory you might be seeing simple as well as the compound microscope. Now here the eyepiece and nose piece, when you people are considering in compound microscope, there 10x, 15x, 20x, like this the x's are written. And say for example the eyepiece and nose piece, they are 10x each. Then the magnification power will be 10x into 10x, that will become 10 into 10, 100. So 100x it is becoming, in the sense whatever the material is present, it will be magnified 100 times. If suppose nose piece and eyepiece, if we are considering 10x and 15x, then 15 into 10 it will become 150. So the material which we are observing, it will be magnified 150 times. So in this way, we are observing these magnifications in microscopes. First of all, in the beginning, Robert Hooke, who observed a cork and he peeled a cork and observed some compartments, that is room-like structures, and he called them as the cellulars, and later it is converted into cell. So now we people are popularly using the word cell. Cell is nothing but a room or it is a compartment. That word we are using in plants and animals. Here each organism is made with the minute structure, what we are calling it as a cell. So such a type of cells, they combine together to form an organism. Say for example, here I am giving nice example. When we are constructing a wall of a building, that time we are using so many number of bricks. The bricks are organized in a proper way, and then it will be sealed, and that total forms a wall. Here the bricks are nothing but cells, and such a type of cells, they combine together to form a tissue. That is, a group of cells, they are forming tissues. A group of tissues, they are forming organs. A group of organs, they are forming organ system. A group of organ system, it is forming an organism. So in this way, the cells are forming organisms. And here Leeuwenhoek, he prepared one of the magnifying glass in 1600s. And during this process, he observed a magnification in the microscope, and first he observed the living cells. And in these living cells, he called them as animalcules. He called these cells as animalcules. And later he observed bacteria, protozoan cells and etc. So in this way the cell study was started. And here the cells what we are studying, that study is called as cytology. Cytology is a study of cells. Now here, when we are taking a cell as an example, already the most important types of cells, that is prokaryotic as well as eukaryotic type of cells you are observing. The prokaryotic type of cells, they are very primitive type of cells. And eukaryotic type of cells, they are advanced type of cells. Now here, the prokaryotic cells, whatever the nucleus they are containing, it is not membrane bound. Membrane is totally absent in the nucleus. And here, the DNA is circular and it is forming the nucleus. We cannot say it as a nucleus-like structure. So we are calling this nucleus-like structure DNA as nucleoid, or that nucleus we are calling it as incipient nucleus. And also we can see ribosomes in case of prokaryotic type of cells. The lower organisms, they are containing such a type of prokaryotic type of cells. In the sense, what you have to remember here is, it is a primitive type of cell. Then eukaryotic cells are advanced type of cells. They are having membrane bound organelles in the cytoplasm and all other characters. So in this way, the differences you people are observing. And also we discussed regarding plant cell and animal cell in earlier clips. There the differences we observed and we studied. Now I am not repeating the same thing. So here we'll move through the questions. Now the first question is, Jesse, Fulgen reaction, a technique has been developed by Fulgen and Rosenbach to study or to stain. So here the options are proteins, lipids, DNA and RNA. So Fulgen technique is used to stain DNAs, that is the genetic material. So the option C, DNA, is the right answer. So option C, DNA. We'll move to the next question. The transmission electron microscope has a resolution of, what kind of resolution this transmission electron microscope is having? We'll see here 2000 nanometers, 200 nanometers, 2 nanometers and 0.2 nanometer. Here the resolution power, that is the transmission electron microscope is having, that is 0.2 nanometers. So option D, 0.2 nanometer is the right answer. So we'll move to the next question. Now see, the electron microscope is used for, why for we are using electron microscope? So electron microscope is highly sophisticated instrument. And here, very minutest material if you are loading, automatically the image will be very bigger. And that image we are using for the cytological studies. Now here, the electron microscope is used for viewing structure of the cell, whole mount study, cell division study, structure of the pollen grain. Here, when materials of pollen grain then cell division, then whole mount you people are observing, okay, the material will be very bigger. So as a result, when you are studying regarding the cell structure, the organelles and other things, they used to be very minute. So because of that here, the electron microscope is used for option A, viewing structure of the cell. So option A is the correct answer here. We'll move to the next. Now see in case of this. Electron microscope has revealed the presence of or which among the following can be seen only under electron microscope. Okay, the options are given here. Which is seen under electron microscope? They ask. So the options are ribosome, chromosome, chloroplast, leucoplast. Here, these are the plastids, and we can see under compound microscope also. Chromosomes are also seen under a compound microscope, but ribosome we cannot see under a compound microscope. So as a result, option A, ribosome, is the right answer here. So we'll move to the next question. Now see, who invented the electron microscope? Who is the founder of electron microscope? So here, Noll and Ruska, Robert Brown, Correns, Johnson and Johnson. So here the right answer is Noll and Ruska. So Noll and Ruska, who found the electron microscope? So option A is the right answer here. We'll move to the next. Now, the part of a cell that can be seen with an electron microscope but never with a light microscope is or the. So the options here are nucleus, Golgi bodies, chloroplast, membrane separating the nucleus from the cytoplasm. So again I am reading the question, just go through it. The part of the cell that can be seen with an electron microscope, but never with the light microscope. Only by electron microscope you can see it, but the light microscope it is not showing it. So the correct answer is option D, membrane separating the nucleus from the cytoplasm. So here we can see only by electron microscope. Nucleus can be seen in the normal microscope, Golgi bodies, chloroplast, everything you can see. But you cannot see membrane separating the nucleus. So, electron microscope helps to see this. So, as a result, option D is the right answer. So, option D is the correct answer. We'll move to the next question. Now, which of the following statements is false? Here, 3 statements are correct, 1 statement is false. So, which one is that? We have to find it out. First one, an angstrom is 100 of a micron. B, separation of proteins can be brought about by electrophoresis. C, the image produced by a compound microscope is virtual and erect. D, unstained living cells and tissues studied under phase contrast microscope. So here all B, C, D are correct options, but here for this question, the false statement is an angstrom is 100 of a micron. That becomes 10 raised to minus 10 micron. Okay. So as a result, this is the false statement. So as a result, option A is the correct one here. We'll move to the next. Now, lignified cells are stained by. Okay, here the cells when you are considering, okay, the cell wall is made with cellulose, hemicellulose, lignin, pectin, you people are knowing. So here, which kind of stain, okay, it will be adapted with the lignin. So what they ask, lignified cells are stained by. Okay, which stain you are using to what it is stain it. So as a result, here first safranin, methyl blue, acetocarmine, light green. So here in cytological staining process, you people are using all these stains. Here safranin is the right answer. Safranin, it actually stains the lignin cells. That is, the cell wall is stained there. So for that purpose, option A is the correct one. We'll move to the next. Now, the smallest size of a cell which can be seen with unaided eye is, or differentiation capacity of human eye. What is the differentiation capacity of human eye, they ask. So here 1 micron, 10 micron, 100 micron, 1000 micron. Here the right answer is option C, 100 micron. 100 micron can be differentiation capacity of the human eye. So option C is the right answer here. We'll move to the next. Now see, ultrastructure of cell can be best studied by, how you are studying the ultrastructure of the cell, okay? Autoradiography, X-ray diffraction method, phase contrast microscope, none of these. Here, ultrastructure of the cell can be best studied by electron microscope. So as a result, here the option D is the right answer for this question, that is none of these. So option D is the correct answer here. Next one. See here, who proposed the cell theory? The cell theory was proposed by whom? We'll go to the option here. Schleiden and Swan, Watson and Crick, Mendel and Morgan, then Robert Hooke. So, if you want the details, you can go through. Robert Hooke, he found the compartments in the cork cell and he called it as a cell. So, Robert Hooke is not right answer. Next one, Mendel and Morgan. Mendel and Morgan, they are related with genetics. So as a result, this is also not a right answer. Now Watson and Crick, if you are taking, okay, they showed a double helical structure of DNA and also they got a Nobel Prize for that. So the Watson and Crick is not a correct answer in the cell theory. But the Schleiden and Swan, okay, who proposed the cell theory? So as a result, here option A is the right answer. Who proposed the cell theory? The cell theory was proposed by Schleiden and Swan. Okay, and also they said the cell exists from pre-existing cell, or sometimes they said the nucleus undergoes budding and that nucleus forms the new cell. So in this way, the cell theory you people are studying, but here the Schleiden and Swan is the right answer. Option A. We'll move to the next question. Now, which of the following is the exception of cell theory? Here, which one is the exception? You have to find it out. So first one is the bacteria. First one, A is bacteria, B is fungi, C is lichen, D is virus. So what they ask, which of the following is the exception of cell theory. Cell theory, see, lichen is containing cell, fungi is containing cell, bacteria is having cell, but virus is all apart from all this. Okay, virus is called living entity. Okay, it is chemical substance. So as a result, here option D is the correct answer for this question. That is the virus. We'll move to the next. Now see here, difference between the prokaryotic and eukaryotic cell in having. Prokaryotes and eukaryotes. Prokaryotes are primitive type of cells, eukaryotes are advanced type of cells. Now here, the cell wall, nuclear membrane, ribosome, none of these. So here, in case of prokaryotes, the membrane bond organelles are totally absent. So here, nucleus is also one of the organelles. Here, membrane bond nucleus is not seen in case of prokaryotes, but it is seen in case of eukaryote. And here, DNA is only the material, that too it is in the circular form found in prokaryote. But here, in case of eukaryote, total membrane bond nucleus you people are getting. So here, the incipient nucleus or false nucleus or nucleoid is seen in prokaryotic type of cell. So as a result, nuclear membrane plays very important role here. So option B is the right answer here. So that is nuclear membrane. We'll move to the next. Intracellular components are not found in cells of intracellular components are not found. Okay, here options: lower plants, prokaryotes, higher plants, eukaryotes. The best answer is the option B, that is prokaryote. We'll move to the next. Now, how many types of cells are known? Here, how many types of cells you people are knowing? So here, usually 3 types. Oh, okay. So option C, 3 types is the correct answer. Now next, the division of the plant kingdom into prokaryota and eukaryota is based on the character of. Okay, and what character prokaryotes and eukaryotes are differentiated? So, see here the options: nucleus only, chromosomes only, cell organelles only, all the above. So, here to differentiate the division of plant kingdom into prokaryota and eukaryota, they are considering all the characters, that is nucleus, chromosome, cell organelles, everything. So, the right answer is option D, all the above. So, all the above is the correct answer. We'll move to the next. Here, the squam and Sclidden were. So, already you studied Sclidden and squam. They are given cell theory. Now, we'll see here. Dutch biologists, English biologists, Austrian biologists, they are German biologists. So, Sclidden and squam are German biologists. So, German biologist is the correct answer. We'll move to the next one. Who proposed the theory that cells arise only from pre-existing cells? So here, the cell theory was given by Sclidden and squam, German biologist, and Virchow improved it a little bit. Okay, and he said the pre-existing cell exists from the older cell or from the nucleus. It undergoes budding, and from that the cell will be arising. So we'll see the options here. Moll, Virchow, then Hickel, Brown. So option B, Virchow is the correct answer. We'll move to the next one. longest cell of plants is found in. The longest cell is found in Victoria amazonica, Eucalyptus, Boimeria nevia, then Sitcoid. So here option C is the correct answer. Boimeria nevia you can take as an example. Next one. The smallest living cells with the cell wall are viroids, algae, bacteria, mycoplasma. Here the bacteria is an example. So option C, bacteria is the correct answer. So dear friends, today regarding cell, the general idea of the cell, what we discussed, some of the questions I presented to you. In coming class, we are continuing the same, and the different organelles we are studying. Apart from that, cell wall, cell membrane, plasma membrane, and apart from that, the protoplasm, cytoplasm, all those things we are studying in detail. And few questions I am selecting from that. Again, the discussion continues. Today, we are completing here and we'll stop it here. Thank you. Thank you.